The type 3 secretion system is a nanomachine that delivers bacterially encoded effector proteins into target eukaryotic host cells. In this electron micrograph, the type 3 secretion system needle complex can be seen at the surface of a gram-negative bacterium. It spans the inner membrane, periplasm, and outer membrane of the bacterium, and a needle extends out from the bacterial surface. A three-dimensional structure of the type 3 secretion needle complex has been generated from high-resolution cryo-electron microscopy images. The base of the complex consists of inner rings that span the bacterial inner membrane, a neck that spans the periplasm, and outer rings that span the bacterial outer membrane. The needle then extends outward from the bacterial surface. In this cross-section, we see a portal through which bacterial effector proteins pass prior to entering the central tunnel that leads towards the host cell. The secretion of bacterial effector proteins occurs when the tip of the needle complex contacts the host cell plasma membrane. Contact triggers secretion of translocator proteins, which form a pore in the host cell membrane. The needle tip attaches to the pore and completes a channel through which bacterially encoded effector proteins pass through the portal, up the central tunnel, and are injected into the host cell. Pathogenic forms of the bacterium E. coli use the type 3 secretion system to inject bacterial effector proteins, including the translocated intimin receptor, or TIR, into a host cell. After injection, the TIR receptor associates with the plasma membrane of the host cell as a transmembrane protein. The extracellular domain of TIR then binds to the bacterial surface protein, intimin. Intimin binding strengthens the attachment between the bacterium and the host cell and enhances the clustering of TIR molecules at the cellular interface. The result is a tight adherence between the bacterium and the host cell. The clustering of TIR proteins stimulates phosphorylation of their tyrosine residues by host cell tyrosine kinases. This leads to the recruitment of actin polymerizing proteins that assemble a branched actin filament network inside the host cell. The polymerization of actin filaments generates a force that projects the host cell plasma membrane outward, thereby forming a pedestal on which the bacterium rests. Here, we see fluorescently labeled enteropathogenic E. coli, or EPEC, labeled green, firmly attached to the extracellular surface of a living host cell and resting on actin pedestals, labeled red. Two of the pedestals are stationary, whereas the other two move, pushing the bacteria across the host cell surface. EPEC is a strain of E. coli that can cause severe diarrheal disease. In this movie, only the actin is fluorescently labeled, revealing the pedestals formed by EPEC bacteria. The bacteria that stimulated pedestal formation cannot be seen, but they are located outside the cell, as indicated here. As the movie loops, note the dynamics of actin in the mobile pedestals. Actin pedestal formation is thought to contribute to the bacterial infection, but the precise role of this remarkable phenomenon in pathogenesis awaits further investigation.